Photosynthesis is a process that converts the light energy into the chemical energy, which is usually referred to the sugars, using the inorganic molecules such as carbon dioxide and water. The process can be conducted by a group of organisms which are photoautotrophs, means that they're able to produce their own food using the light energy. Photoautotrophs can be the unicellular protists such as Euglena species and the algae, and also the higher class of plants such as the angiosperms. The photosynthesis process can be represented by the equation below, where the carbon dioxide molecule will combine with the water molecule to produce the sugar, which is the glucose, and oxygen as the byproduct. With the presence of light energy, such as the sunlight, and this process occurs in chloroplast, where the chlorophyll pigment is found. There are two stages of photosynthesis which occurs at the chloroplast. Photosynthesis begins with light-dependent reaction, which takes place at the thylakoid, and followed by the light-independent reactions, which is also known as Calvin cycle. This will occur at stroma of the chloroplast. Chloroplast is the double membrane organelles that refer as the site of photosynthesis, and these organelles are found at the cytoplasm of any photosynthetic cell, such as the plant cell. And we have to study the structure of chloroplast that relates with the stages of photosynthesis, which are light-dependent reaction and light-independent reaction. Chloroplast is an oval disc-shaped organelles which are enclosed with two layers of plasma membrane. The outer membrane and the inner membrane are both smooth. The inner membrane of chloroplast encloses a space which is fluid-filled, called as stroma, where is the site of light-independent reaction. In the stroma, there are the structure called as thylakoid, which is a membranous structure, and they are arranged in a step form to form granum. This arrangement will save space to store more thylakoid, and each thylakoid will be the site of light-dependent reaction. This is why they are able to provide more surface area for light-dependent reaction. So a group of thylakoid, they will stack to each other to form a granum. And each thylakoid, they are membranous structure with a space within it. We call it as thylakoid space. At the membrane of the thylakoid, there is a group of protein complexes. They will form photosystem, which is important in the light-dependent reaction. At the membrane of thylakoid, there is a group of protein complexes we call it as photosynthetic pigments, such as chlorophyll B and chlorophyll A. These pigments, they are able to absorb light energy or photon. Different types of the pigments able to absorb different wavelength of light. So most of the chlorophyll or most of the photosynthetic pigments, they are going to refract the green light. This is why we observe the chloroplast in green under microscope. There are two types of photosynthetic pigments, which are primary pigments and accessory pigments. Primary pigments consist of chlorophyll A, which have two types, Photosystem 680 and Photosystem 700. Both are important in the process of light-dependent reaction called as photoexcitation. And primary pigments are helped by accessory pigments, which consist of chlorophyll A, B, carotenoids, which include carotene, xanthophyll, and phyophytin. Each of these accessory pigments, they have different color because they're able to refract different wavelength of light. At the same time, they're able to absorb different wavelength of light. This is why accessory pigments absorb the sunlight for greater range of wavelength, because we have many types of accessory pigments here. After they absorb the sunlight, they are going to transfer this absorbed photon or light energy to chlorophyll A for the process photoexcitation. During the absorption and transferring of the light energy to chlorophyll A, they able to lower down the light energy. So they protect the chlorophyll A from the destruction by excessive light energy. Scientists has identified the most effective wavelength of light, which means the specific wavelength of light that able to promote photosynthesis. So if we study the x-axis here, they are consist of the visible light from the violet, blue, green, yellow, orange, and the red color, which is located from the shortest wavelength 400 nanometer to around the 700 nanometer. So if we study the examples of chlorophyll A and chlorophyll B, which is the pigment found a lot in the plant cell, they absorb best in the blue and red regions, because we notice they achieve the highest value of light absorption when these chlorophyll are exposed to blue light and the red light. However, carotenoids include the carotene, xanthophyll, and phyophytin. They perform the highest value when they are exposed to the violet light, because the value is the highest here, and also the blue-green region. And after that, the graph show very low value. That's mean no absorption at the region after the blue-green light. And after that, they study the plant cells activity in terms of photosynthesis. They measure the oxygen released by the plant cell when they are exposed to different wavelength of light. And this graph we call it as action spectrum, because we study the action of the pigments in terms of photosynthesis. And then we notice that when the plant cells are exposed to the violet blue wavelength and the red wavelength, the oxygen released from the plant cells is the highest value. This is why they say that these two wavelengths of light are the most effective light to promote photosynthesis. Both photosystem 1 and photosystem 2, they have the components consist of reaction center complex and also the light harvesting complex. The name have the suffix complex because this group of proteins, they are consist of more than one unit, which is a large size of molecules can be found in the membrane of the thylakoid. And the reaction center complex usually consists of a pair of chlorophyll A, which is the primary pigment. And the main function of these chlorophyll A molecules, they are going to become the primary electron acceptor. However, the light harvesting complex, if you refer to this schematic diagram, as I say, they are surround the reaction center complex. and they are consist of accessory pigments that we have studied before, which include the chlorophyll A, chlorophyll B, and carotenoids. Why they are called as light harvesting? Because they are going to capture the light energy or the photon, which means that they're able to absorb the light energy, and each pigments they have different wavelength of light absorption. After they absorb the light energy, they transfer the photon from pigment by pigment until they able to be absorbed by the reaction center. So with the absorption of light energy at the reaction center, a process we call as photoexcitation happens, which is symbolized by the increased energy level of the electron. In the previous slide, we already focused on one of the important structure of chloroplast is thylakoid. Thylakoid is a structure that enclosed by membrane. And at the thylakoid membrane, there are a group of photosynthetic pigments. They always cluster together to function as photosystem. The name implies that these photosynthetic pigment, they are going to become a light capturing unit. So there are two types of photosystem can be found in the chloroplast or at the thylakoid membrane, which are photosystem 1 with the abbreviation singkatan dia PS1. The one must be written in Roman. The next photosystem is photosystem 2 with the abbreviation PS2. These two photosystems show a few differences. The main one is the reaction center that consists of the primary pigment, which is chlorophyll A, is different. Photosystem 1 has chlorophyll A P700 and photosystem 2 has chlorophyll A P680, which is function as the reaction center. However, the name is given because depends on the wavelength of light can be absorbed by chlorophyll A. For photosystem 1, the chlorophyll A able to absorb the best at 700 nanometer, which is actually the far red part of the light spectrum. And photosystem 2, the chlorophyll A absorb the best at 680 nanometer, which is actually the red part of light spectrum. Both photosystems are important in a process called as photophosphorylation, which means a ATP synthesis occurs due to the light energy. And PS1 will involve in both cyclic and non-cyclic photophosphorylation, and photosystem 2 only involve in cyclic photophosphorylation, and these both cyclic and non-cyclic we are going to study more in second hour. Okay, the main function of photosystem 1 actually at the end of the photophosphorylation process, they're able to produce ATP and also the electron carrier, which is high energy molecule also called as NADPH. And photosystem 2, they're able to produce ATP also since that they perform cyclic photophosphorylation, but they do not produce an ATPH. They will show another significant process, which is photolysis of water. That means the water molecule is splitted by light energy. So in the following hours, we are going to study the two stages of photosynthesis in more details. So photosynthesis will begin with light-dependent reaction first. And they require the inorganic molecules, which are water, that able to be splitted when they undergo a process called as photolysis of water. Through this process, oxygen molecule can be produced. So oxygen actually is the byproduct from the light-dependent reaction. And light-dependent reaction required a photon because it is absorbed by photosystem 1 or photosystem 2, or it can be absorbed by both photosystems at the same time to perform different types of ATP synthesis. First is non-cyclic photophosphorylation, which is a type of ATP synthesis happens in the photosynthetic cell. And there's another pathway to produce ATP, which is cyclic photophosphorylation. The difference is non-cyclic photophosphorylation, they're able to produce the high energy molecule, which are NADPH, which is also the electron carrier. So, jika tengok nama dia, molekul ini sebenarnya dia lebih mirip kepada kita punya NADH dalam topik cellular respiration. Tetapi disebabkan sekarang kita belajar photosynthesis, that's why the electron carrier, the structure is different with the NADH in cellular respiration. The name is NADPH. Okay, this product from the photophosphorylation, actually they are being used in the light-independent reaction, which is another stage of photosynthesis. So when the ATP and NADPH are used by the light-independent reaction, which is also called as Calvin cycle, they are going to produce the sugar from inorganic molecules, which is the CO2. Okay. So through the process of Calvin cycle, actually they are produced the NADP plus and ATP. So it seems like recycle back the molecule to be used in the light-dependent reaction. This is the relationship between light-dependent and light-independent reactions. Both stages occurs at organelles, chloroplast, but the specific location is still different. Light-dependent reaction happens at the thylakoid membrane. Light-independent reaction occurs at the stroma.